now. I think we should be starting now. Okay, it's a great pleasure to have Romain Levayer from France Pasteur Institute. Romain did PhD with Thomas Lequi in Masa, primarily focusing on epithelium morphogenesis during germ blood extension. And then since then, he moved to Switzerland for postdoc to work with Edward Moreno to change the topic into cell composition. But having a strong background in morphogenesis, Romain established a very nice concept of mechanical cell competition. And then he had a brilliant publication. Then he came back to Paris, and he's been having own lab for several years. And so he is still interested in collective cell behavior underlying morphogenesis and cell competition or cell elimination. Okay, Romain.
All right, thank you, Masa, for the introduction. Yeah, it's been already 5 years I've been in Paris, actually, time is flying. I will start sharing my screen, and I hope I will still be able to see you properly. And of course, if there is something going wrong and if you can't hear me well, or if you have a question, just turn on your microphone and make some noise and I will interrupt. So yeah, it's a real pleasure to be here virtually. I would have loved to come in person, but I guess, well, you have restriction, and even in my campus, it's very difficult to have people coming. We have no in-person seminar right now, so that would have been weird to come in person, but maybe another time. And so what I would like to tell you about today is a different project that we've been working on lately in the lab. Some of them have been published very recently, which deals with the fine-tuning of cell death in epithelial tissue. So you will see there will be a link with cell competition, but there is also a very important aspect related to homeostasis of tissue. Now, if I have to introduce the key question and the global question we are interested in my group, in a way, it's all related to the plasticity of developing tissue. Because by plasticity, I mean their incredible capacity to cope with massive perturbation, and yet developed a structure which is totally fine in terms of organization, in terms of cell type distribution, and in terms of shape as well. And of course, the most extreme example of plasticity come from this organism, which have an incredible regenerative capacity, like the flatworm here, which you can cut in 64 small pieces and will form 64 little worms, all perfectly well shaped, very bit smaller, but they have all the right cell type at the right place. And actually, this type of self-organizing property is very intriguing. And in a way, this is all about trying to understand how the cell, from its perspective, take the right decision. You have the cell in the middle of a tissue, there will be some perturbation. And then the question is, what are the kind of information that the cell is probing to take the right decision and adapt to the perturbation? Could it be cell multiplication, cell death, fate specification, or cell movement, for instance? And so, of course, these are the framing of very big question, but in the lab, we are not working on the flatworm. Maybe we should, but we don't. We work on Drosophila, and we are not working on any type of cell decision. We are focusing on one specific type of decision, which is cell death and apoptosis in epithelial context. And actually, I would argue that we know quite a lot about apoptosis pathway, but when you place back apoptosis in a tissular context, there are many big questions which we don't understand at all right now. And here I list some of those questions, which are kind of a long lifetime goals, because most of them are very challenging. So what I want to frame here is the fact that you have a tissue here where you have cells dying in red and cell dividing in blue. And in many instances during development or homeostasis, we don't really know what is exactly the distribution in time and space of cell elimination. Moreover, even if we knew this distribution, we don't really know what is regulating the precise distribution in space and time of cell elimination. Then a key question here is, how does the system count? How does the system know there is 20% of cell dying, not 40% of cells, which is a bit related to the question of size regulation in tissue. And the same applies for cell death regulation. We don't really know what sets the number of cells that will die in a tissue. Then a related question is, how plastic is this process? Is it very much cell autonomous? You have a kind of constant rate of cell death and then everything happens randomly? Or is there some sort of coordination between cells that can help to readjust the rate of cell elimination? So for instance, if I block cell death in half of the tissue, what happens here for the remaining part? Is there some sort of adjustment? At which range and how much is it plastic? Now, this you can you can have this question in homeostatic process, but this is also relevant in perturbed condition, which I will describe in a minute, which is related to cell competition, which is one illustration of this plasticity where some cell type can co-opt the elimination of the neighbors and take over in the tissue. Now, those are all questions related to what happened upstream of apoptosis induction, but we are very much interested by what happened downstream, which is, for instance, how does the cell commit in apoptosis? We know that there are many cells which activate caspase and yet don't die, and we are trying to understand how eventually the cell commits in cell death. And then how do you get rid of the cell from the epithelial layer, which we name cell extrusion? So what are the remodeling steps that kick the cell out of the tissue layer? And so today, obviously, I will not cover this full menu because that will last for hours. And for many of these questions, we don't have answers. But I will cover those three aspects here. So I will first briefly describe some results we had regarding cell competition and introduce the concept. Then I will try to show you some recent results about the distribution of cell death in time and space. And I will finish with some recent results regarding the regulation of cell extrusion in Drosophila. Now, when it comes to cell competition, I would do a very brief introduction because I guess many of you heard about it from the Institute. So cell competition is a field that emerged, well, more than 40 years ago, but started to really explode 10 years ago, I would say. And if I have to find a definition of cell competition, which is still a bit debated, the simplest one is the fact that this is a context-dependent cell elimination process. So that means that in a way, if you have cells here which are totally viable, but it's only when they are surrounded by another cell type that those cells will be eliminated either by extrusion or by apoptosis. And so this was characterized more than 40 years ago in fly, in Drosophila, using a bunch of mutation called minute, which reduced the quantity of ribosomes in cells. And basically what is quite fascinating is that the minute flies are totally fine, there is no defect. They tend to grow slower, but they reach the adult stage and there is no visible defect. However, when you start to generate mosaic tissue with minute cells surrounded by wild-type cells, then those cells will be eliminated from the growing tissue and you will not recover those cells in the adult. And which was shown later on to be based on apoptosis. And so the key question is, what is driving this context-dependent apoptosis? Now, the field really started to explode a bit more than 10 years ago now, where it was found that overmutation will somehow increase the fitness of cell, which will start to eliminate the neighboring wild-type cell and take over the space in the tissue. So this was found, for instance, for MYC, a famous oncogene. If you increase the number of copies of MYC, then those cells will eliminate the wild-type cell and replace them in the tissue. And the same apply for other growth factor like wingless or YAP and Yorkie, which can also trigger the same process, which obviously make a connection with the early phase of tumor progression and drive a lot of excitement for the field of cell competition. And so nowadays we have hundreds of examples of cell competition with different genetic background. It is quite, it has been observed in many different tissues, from the fly system to mammals in the skin during early mice embryogenesis. And one big key question, which is not really yet totally understood, is what is the mechanism behind? How does the system recognize another cell type and trigger its elimination? And one of the reasons is that because most likely there are different mechanisms that can act together to eliminate the cell. And to make a long story short, you can summarize the main models in three main categories. On one hand, there has been system where it has been proposed that there is a fight for some survival factor, which would be in limited quantity. So in that case, the purple cells will bind all the survival factors, which will drive the elimination of these green cells, which we call loser cell. In other systems, what has been proposed is that there is a specific ligand receptor interaction at the margin between the two cell types, which will trigger cell elimination here at the boundary. And the last category, which I will talk more about today, is related to mechanical stress, with the idea that if one cell type is more sensitive to mechanical stress, this one will preferentially die in the tissue. So I will spend a bit more time on this one because this is something that we've been studying a bit lately and we are still working on right now. And actually, this idea came quite a while ago by the work of Boris Romain, a theoretician in Santa Barbara, who published a paper on this in 2005, who proposed a rather simple description of a competitive process where you would have two cell populations that grow at different rates. And because the tissue behaves like a solid, this differential growth rate would lead to the buildup of pressure and the accumulation of mechanical stress in the tissue. Now, if on the top of this, you assume that one cell type, here the blue cell type, is more sensitive to pressure, then this one will die preferentially, and in the end, you will completely replace the blue cells by the green cell. So this was a very nice theory. And actually, there is another theory that came out a few years later, which is based on homeostatic pressure. So it's a bit different because here you assume more like a gas description of a tissue. And I will not speak about that, but there are very good examples of experiments that complement this model, which partially the work of Guillaume Charras here in the audience. But so focusing on Boris Schreiman framework, for some reason, this was not so much studied experimentally. And I think one of the reasons is that there are many hypotheses behind which were not really fulfilled at the time. So first, you need to show that differential growth would lead to the accumulation of mechanical stress, which means that the tissue on the timescale of growth have to behave more or less like a solid with very little cell movement. On the top of this, you have to prove somehow that compaction of cells can drive some cell death and cell elimination. And you need to find a parameter that can explain why different cell type would have a differential sensitivity to compaction. And so in a way, we came into this because we found a system where all these hypotheses could be fulfilled, which gave the proof of principle that we could have such mechanical competition. And so the system we use, which I will be describing mostly in this in the seminar today, is the pupal notum, which is a single layer epithelium at the back of the fly at the pupal stage, which you can see here, marked with cadherin GFP. And all the little dots that you see here are marking cells which are extruding and dying from this epithelial layer in the 24 hours that you see here, which means that there is a fairly high rate of cell elimination here. There are more than 1,000 cells dying in less than 24 hours. And you can see a little close-up view here. Those cells start to extrude. They are contracting the apical area. They are expelled from the tissue and they fragment in small pieces. So we use this system, and what was found by S and others, actually, to make a long story short, is that the cells in this system are sensitive to deformation. So this was based on the pioneer work of the group of Busbaum, actually, which we confirmed later on. We know that if we stretch the cell, we promote the survival of cells, and if you compact the cells, you can promote the cell elimination. So what we had here in this layer is the fact that the compaction will directly impact caspase reactivation, which will lead to cell extrusion and cell death. And in a way, based on this very simple diagram here, we have now all the ingredients for mechanical competition because we know that compaction can trigger cell death. And basically, if you modulate the capacity of the cell to activate caspase by blocking apoptosis or blocking caspase activation, you will block the sensitivity to compaction. So we have a way to modulate the sensitivity to compaction and therefore potentially a way to trigger this mechanical competition. And so as a proof of principle, this is exactly what we see here when we activate the oncogene RAS in a large group of cells, which you can see here in magenta. So those cells where we activate RAS becomes bigger and bigger. Then you will start to see some deformation in the neighboring cells, which are becoming smaller and smaller. And then what you see in white is ectopic cell elimination through apoptosis in a region where normally you hardly see any cell elimination. So that was the proof of principle that yes, we can trigger some ectopic cell death based on the overgrowth of this guy, which triggers some deformation and triggers some cell elimination. And something you should know is that those RAS cells are highly resistant to apoptosis because they inhibit one of the main pro-apoptotic genes in this system, which is important for cell death induction. Now, this is a proof of principle. And of course, as usual, this opens many questions, which we are still trying to address now in the lab. So the first one is, at the time, we didn't know what were the pathways sensitive to deformation and triggering cell death, which is a typical question of mechanotransduction. And the other question, which I will not describe today, is what are the minimal conditions allowing such mechanical competition? Is it true for any sort of oncogene that promote growth? Do you need a certain clone size or clone shape to have this kind of elimination? So can we generalize this process? And we are actually studying on this thing right now. And what I can say is that we see that depending on the oncogenes, there are different processes that can participate to the compaction, including also some change of the mechanics at the boundary between the two cell types. But I will not talk about this today. What I would like to briefly mention now is the first part here about the pathway that sense deformation and trigger cell death. And so to make a long story short, because this we published a couple of years ago, we tried many pathways until we bumped into one famous pathway, which is the EGFR RAS ERK pathway, which is a very important pro-survival pathway in Drosophila and also in the pupal notum. So this is mostly going through the inhibition of this gene here, which is a pro-apoptotic gene. And we know that EGFR is essential for survival in the notum. And basically, you need to downregulate EGFR to see a local increase of cell death. But then the key question we had is whether this guy could be sensitive to deformation. And we were stuck for a while because we didn't have a way to correlate deformation with the variation of ERK signaling, because there was no proper live sensor of ERK signaling pathway. And that's why we developed one, which is a rather simple one. So that's what we call mini capicula. So it's a minimal domain of a substrate of ERK, which shuttle from the nucleus to the cytoplasm. Upon phosphorylation, you exclude the fluorophore from the nucleus. And this way you can locate in live the activity of ERK at a single cell level. So it's very similar to this KTR construct that you probably heard about, which are now widely used in mammalian system, except that here it's based on a kind of minimal domain from a substrate in flight. And so this system works fairly well. I can show you one movie here. We have this quite complex dynamics with global activation and inhibition. But what you can see here is some domain like here in the posterior domain and in the midline, where there is this high nuclear signal, which means low ERK signaling. Keep that in mind, it's a bit contradictory. When you see a lot of signal, that means there is low ERK activity. And actually in this region, there is a lot of cell death, and it correlates perfectly with the place where there is more cell death. Now, for the sake of time, I don't want to present the result here, but what we found is that indeed, if we stretch the tissue, we can activate ERK. If we release tension in the tissue, we can downregulate ERK. And this is actually exactly what's happening in the neighborhood of those clones that overgrow. So we see a downregulation of ERK that triggers cell elimination. And so if you want to know more, this was published a couple of years ago, because I'd like to focus more about more recent projects related to this. So basically, we know that ERK here is sensitive to deformation. When you have more tension, more stretching of cells, there is an activation, which is pro-survival. When you compact the cells, you downregulate ERK, which promotes cell death, which happens near the RAS clone that overgrow. Now, again, these are the conclusion, but that opened many questions, including questions that we are still trying to solve nowadays in the lab. So the first one is we don't really know what's the mechanism of mechanosensation. So what is the link between cell deformation and the modulation of the EGFR pathway? So we have some clues. We've been trying for 3 years to test many models, failing one after the other, and we are still trying to find a mechanism. But yeah, this is still something that we don't quite understand. The other question which I would like to talk about more today is what is the physiological function of the link between mechanics and modulation of ERK signaling? Because so far, what I've been describing here was mostly based on very artificial condition, where we trigger overgrowth in a clone, we trigger some laser perturbation, but whether the modulation of ERK in physiological context by mechanics is important for development was not clear at this stage. And so the project I would like to present now actually give us some hint about the putative function of ERK mechanosensing in the homeostasis of tissue. So this is based on the work of Leo, one of the first postdocs who joined the lab, who now got a permanent position in the lab, actually. And then we touched this question by addressing a completely different question, which was the question of how can a tissue deal with very high rate of cell elimination? Because something that may not seem so obvious is that there are many systems during development where there is a massive elimination of cells in epithelial layer, and yet you can maintain the cohesion of tissue at any time, which is very surprising because we know that cell extrusion can help to expel a single cell, but what happens now if you have hundreds of cells dying simultaneously? And so just to show you an example here, this is a movie from the abdomen of the fly, where you have those gigantic larval cells that will be all eliminated within 24 hours and completely replaced by this guy here. And yet the cohesion is maintained during all the process. So how is it possible? Is there any rules regarding cell elimination to make sure that you can maintain the cohesion of the tissue? So that's the question we start to address in the pupal notum, because in the notum, there are thousands of cells dying. And first, to address this question, we first start using perturbative approach by forcing cell death using a new optogenetic tool. So what we did is we developed an optogenetic tool to activate caspase, where we fuse DRONC, which is the Drosophila caspase 9 here, with this Cry2 system, which oligomerize upon blue light exposure. And by oligomerizing the caspase 9, you will trigger the cleavage of the caspase, which then trigger apoptosis induction, which works very well in the notum. In less than half an hour, you can kill most of the cells which are expressing the tool here. Now, what Leo did is expressing this tool in different numbers of cells, 1 cell, 2 cells, 3 cells, 4 cells, using a clonal expression, and try to see when we start to see aberrant cell extrusion in the system, testing the limit regarding concomitant cell extrusion, actually. So this is what we see here. We eliminate 1 cell, 2 cells, 3 cells in line, 3 cells in cluster, 4 cells in cluster. And so what Leo could see is as long as cells are organized in lines like this, extrusion occurs fine. There is no visible defect during the extrusion process. However, as soon as we start looking at clusters of cell elimination like here, what he could see is some sort of aberrant cell extrusion. which you can focus on here. So what you see is that the cell at the beginning will start to constrict. It will play again. So those cells here will start to constrict. Then boom, there is a relaxation phase. Then you lose cell adhesion between cells. There is some sort of gap formation, which is followed then by wound healing and closure of the gap, which is very prevalent as soon as we have these clusters of cell elimination. And I will show you the snapshot. The cells start to constrict, but then somehow there is this relaxation phase where we lose adhesive component between cells. Then this is followed by a typical figure of wound healing with this accumulation of cadherin at tricellular junction and the closure of the gap. So that suggests that eliminating cell in cluster is very problematic for the tissue, because this seems to suggest that there you transiently lose the sealing property. And indeed, we know we lose the sealing property because if we inject dextran in the tissue, if you eliminate cells in line like here, there is no leakage of dextran between cells. However, as soon as you eliminate cell in cluster like this one, every time we see those sort of aberrant cell extrusion, this is concomitant with a leakage of dextran between cells, which is transient. Then, of course, the gap gets closed. But obviously, if this occurs repetitively, this could endanger the tissue on the long term. So there is something quite interesting, because as little as 3 cells in cluster that are eliminated together seems to be very bad for the tissue. And if you do some basic calculation, we would expect to see such cluster in the notum, because there is a very high rate of cell elimination, and the chance to have 3 cells next to one another is different from 0. We should see some clusters, and yet we never see them, which is suggesting that there has to be something regarding the regulation of cell death distribution. And for this reason, Leo started to focus more on the distribution in space and time of cell elimination in the pupal notum. So what it does is every time is basically locating every single cell death in the notum, which you can see here with the colored dots. And just to be clear, if I now superimpose all cell death, the pattern looks like this. We are not interested here by the global tissue pattern, but more focusing here on a specific region, trying to see if there is any rules regarding the precise distribution in time and space of cell elimination. And so we focus here because this is more or less flat in X, Y, and in time. And then we use a certain number of techniques to see whether there was any bias in the distribution. Is it like a Poisson process, where every cell death is independent, there is no coordination between cells? Is there some clustering effect, or is there some dispersive effect that tend to separate cell death in time and space? And so we did plenty of analysis. I will just show you the result of one type of analysis, which is using this K-function dispersion map. So the K-function calculates the deviation in the distribution compared to a Poisson process with an equivalent rate. So that means that if you are totally blue, then it means the distribution perfectly matched with a Poisson process where everything is independent and totally random. If you see here some yellowish color, it means that there is a significant dispersion in the data compared to a Poisson process, which means there is less death than expected if it would be a random process. And this is exactly what we see here. So this is the time distance and the spatial distance. You see the yellow color here, which means that every time a cell dies, for the next hour, and at a distance of 10 micrometer, there is less death than expected if everything would be random. And indeed, you can exactly reproduce the same sort of distribution by simulating a Poisson process where you had a refractory phase that lasts roughly 40 minutes at a 5 to 10 micrometer distance, which corresponds to 1 cell diameter, basically. So to summarize this, and this was based on several analyses actually, we found that cell death distribution in the notum does not follow a Poisson process locally. There are some bias in the distribution, which is characterized by the fact that every time a cell dies, there is a transient and local inhibitory phase on roughly 1 cell distance that inhibits cell death for 1 hour. So there is a reduction of a factor of 2 of the probability to see cell death for 1 hour. So the next obvious question is, what is responsible for this refractory phase? And so the first thing we check is caspase activity, because we know that caspase activation precedes cell extrusion in the system and is one of the key regulators of cell elimination in the pupal notum. And so here what you have is a live marker of caspase, effector caspase, based on a GFP sensor, which becomes fluorescent upon cleavage. And what we notice is that there are many situations where we have clusters of cells that start to activate caspase altogether simultaneously, but then there is one cell dying and the other cell remain in the tissue and keep a constant GFP value. So this one will die here, and then this one will reach a kind of plateau value and will stay in the tissue for hours and will not die actually. And this is exactly what we see here in terms of intensity. The green one is the one dying, the magenta one is the one that reached this plateau, and the one that will remain in the tissue. Now there is a little trick here because the GFP is very stable. It is stable over hours here, which means that what is really relevant regarding caspase activity is the rate of accumulation of the GFP signal, which means that in that case, caspase activity is on But then because you reach a plateau, then caspase activity go back to zero level or steady state level. So that means that there is a transient, or there is some sort of inhibition of caspase that occur near the dying cell here, which is exactly what we see on this average curve. So here, this is based on several cells. You have one first cell that die, and in the neighbors, there is a transient activation, which is then followed by inhibition and cell going back to the steady state level. So there seems to be some kind of caspase inhibition triggered by cell extrusion. And the next question was, what is upstream of this caspase inhibition? And as you may already guess, that's where we made the connection with the previous project, because we looked at ERK signaling pathway, as we knew it was a central regulator of cell survival. So what we did is this kind of large-scale movie where you have cadherin signal, a marker of nucleus with histone here, and the marker of the ERK signaling pathway. And so what you can focus on here is the posterior region here, where you have all this nuclear signal, which means low ERK activity. And what Leo could notice is this kind of sporadic clouds that appear here and there, where we lose the nuclear signal of the ERK sensor. And at the beginning, we thought it was cell division, so we didn't pay attention until we realized it was not cell division, but actually cell death. So here you have a dying cell in the middle, and then boom, in the neighbors, you lose the nuclear signal of the sensor, which means that there is a pulse of ERK activity. And you can see exactly the same with the threat sensor here. The cell is dying, and then boom, you see this reddish color, which means that there is an activation of ERK in the direct neighbors. And so we did a lot of characterization of this pulse, and I would just sum up what we found. So we found that this transient ERK activation systematically occurs near the dying cell and is restricted to the direct neighbors of the dying cell. We don't see it propagating to several cell rows. It correlates very well with the cell stretching, because during extrusion, those cells become larger, and then this correlates perfectly in time with the activation of ERK. More importantly, we can even mimic the process by laser ablating a single cell here. This will generate some stretching and also some activation of ERK. Although I should say there is no definitive proof that it's purely mechanical. We've been excluding the different mechanism one by one, but it's very difficult to definitely prove that this is purely mediated by cell deformation. And then most importantly, those feedback are based on EGFR. If you remove EGFR, even in clones, you completely abolish this pulse of ERK activity. So then the question is, can we relate this EGFR ERK pulse with the distribution of cell death? So again, I will not show you the raw data, just go straight to the conclusion. But first, we found that the pulse of ERK systematically precedes this inhibitory phase of caspase with a lag time of 15 minutes. So you see first ERK pulse, and then 15 minutes later, you see an inflection in the caspase activity curve. Then what we did is we checked whether EGFR was important for those change of caspase activity. And indeed, what we found is that when we deplete EGF receptor, we don't see anymore these caspase inhibition events in the neighboring cells. So the next obvious question based on this was, can we see any change in cell death distribution in this context? So what Leo did is systematically, again, locating cell death in condition where we deplete EGF receptor in a large portion of the tissue. So there is some ectopic cell death in that case, but interestingly, in the posterior region, there is not such a massive increase of the rate of cell death, most likely because we start with very low basal ERK activity here. So then he analyzed there the distribution using the same sort of tool. And I would not I would not show you all the different sort of analysis. I will just show you this K-function analysis again. So as I showed you at the beginning, in the wild-type context, there is this significant dispersive effect in the wild-type situation. When we deplete EGF receptor, we completely lose this dispersive effect, suggesting that now the distribution becomes random like a Poisson process. And as I mentioned at the beginning, if this is a random process, there should be some clusters of cell elimination that occur spontaneously. And this is exactly what we saw in the EGFR context. There are many cases where we see this kind of clusters of cells. Here you have 4 cells dying simultaneously. This is followed by this aberrant extrusion, where there seems to be some kind of gap formation, followed by wind filling. And this is the quantification here. In the wild-type context, we almost never see such clusters. When we deplete EGF receptor, this becomes very likely. There is close to 4 or 5 clusters observed on each movie, which of course repetitively can endanger the tissue. And we even see sometimes some tissue collapse, although it's quite rare, but this happens sometimes. So to sum up what I show here in this project, what I show here is that there is this very interesting local feedback that occurs near every extruding cell. So every time a cell dies, there is a transient activation of ERK in the neighbors, which inhibit locally caspase activity and help to distribute in time and space cell death to make it more dispersed in the tissue. And this is very important to have such feedback in condition of high rate of elimination, because in absence of the feedback, then we have clusters of cell elimination that occur, which are associated with transient loss of sealing property, which of course on the long term could be very damaging for the tissue. And what I like here about this conclusion is that it reflects the very interesting self-organizing property of an epithelium, where the robustness actually emerged from very local interaction between cells. I'm not talking about some sort of large-scale integral feedback. I'm talking about a very local communication between cell and its neighbors associated with a very transient activation of ERK. And yet at the tissue level, this is essential to maintain the cohesion if there is a high rate of cell elimination. And more importantly, this is not a peculiarity of the fly system, because a very similar process was described in a mammalian system by the group of Olivier Pertz in Switzerland at the same time. So here, this is in MCF10A and MCK cells. You have a dying cells here, and then boom, you see this loose of nuclear sensor. It's the KTR sensor of the ERK pathway, which means that there is a wave of ERK activity in the neighbors every time a cell dies. So it's a bit different from the fly system, because there it propagates on 2 to 3 cell row, and it has a very nice buffering effect on the global rate of cell elimination in the tissue if you impose any sort of stress that trigger apoptosis. So that means that this kind of protective effect seems to be conserved on a rather large scale in evolution. Now, I would like to finish on this part with a few speculative points, because I've been describing cell competition, then I've been describing this physiological process that helps to distribute cell elimination in time and space. And then the question is there any connection between the two processes? And actually, I do believe that what we show here may be very relevant for cell competition for 2 reasons. The first thing is this kind of movie that I show you here, where we have clusters of cells that activate caspase, and then there is one cell die and the other remain in the tissue, suggests that cell death is not at all cell autonomous. There is a high non-cell autonomous effect here, and which is very similar to lateral inhibition and notch signaling pathway, for instance. So you have one first cell that die, and this inhibits cell death in the neighbors. And so because there is a tug of war between cells regarding who's going to die and who's going to stay in the tissue, there is room here for some sort of selection process that could prime a cell for dying first, and therefore being preferentially eliminated in a tissue, just like what you would expect from cell competition. And the other aspect which I would like to present here, which is right now purely theoretical, is the fact that the same process could help to redistribute cell death in a tissue. So imagine you have a clone of cells here, which are resistant for apoptosis. That means that the guy here neighboring the apoptosis resistant cells will almost never have ERK pulse, because there will never be a cell dying in the neighborhood. However, this one, because there are wild type cells around, there will be quite often cells dying around, so it will receive several ERK pulse during the duration of the movie. So effectively, that means that this one should have a higher chance to die compared to this one. And this is exactly what we see if we simulate this, provided you have a high rate of cell elimination. Having several neighbors resistant for apoptosis should be enough to increase your chance of dying. So this is exactly what we see in the classic cell competition scenario, where you have one clone that trigger apoptosis in the direct neighborhood of the clone. And that could be based just on this kind of imbalance in the feedback that could help to redistribute cell death. And now what we are doing is testing whether this is true experimentally. Okay, so again, I'm kind of stressing the message, but the philosophy we have in the group, and which has been very fruitful so far, is that we go back and forth between the cell competition, which are kind of a pathological situation, which help us to identify pathway, like the ERK pathway, for instance, which happen to be very relevant for homeostatic process and tissue plasticity. And then this kind of process that we describe in homeostasis may be very relevant for cell competition. And I have a strong belief that this kind of back and forth movement is very essential if we really want to understand fully the mechanism of cell competition and its physiological function. Now, I will, maybe for the last less than 10 minutes, actually, finish with a different scale of study. So I've been describing so far things related to regulation of apoptosis and tissue plasticity. Now I would like to focus on the last minute on what happened downstream of caspase activation in the epithelial layer. So there are 2 questions here. So how does the cell commit in apoptosis, and how do you regulate cell extrusion? So I would like to play again this type of movie where we have a sensor of caspase activity here in green. And what you have here encoded is the live caspase activity, so the rate of change of the GFP signal. And as you can see here in this movie, we have many cells which activate caspase and yet will not die during the process. And there are many cells that transiently activate caspase and do not die. And the big question here is, what is the decision point? How can you predict whether a cell will die or not downstream of caspase activity? And this is an essential question which we are starting to address in the lab, trying to see if we can pinpoint or identify some parameters that can help to predict which cell is going to die versus which cell is not going to die. And I'm not going to talk about this because this is something that we just taught quite recently, but this is becoming something very important in the lab. Now, the other thing I would like to speak more today about is how then do you regulate cell extrusion downstream of caspase activation? So how do you expose the cell, and how does caspase orchestrate the extrusion of cell from the epithelium? And so this is the work of Alexis Villa, a PhD student who's now finishing this year, actually, who will be soon on the job market. And so the question here is, we know that caspase activation always precedes cell extrusion and is essential for cell extrusion, but how does it regulate the extrusion process? So to summarize the work from many groups, including actually the group of Guillaume, we know quite a bit about cell extrusion, mostly from MDCK cells and a little bit from zebrafish and Drosophila as well, and zebrafish. And if I have to sum up the work from many labs, the main regulator of cell extrusion is the formation of an actomyosin cable, which is formed in the dying cell and the neighbors, which will constrict the cell and expose the cell from the tissue layer. Then at the same time, there is a disassembly of cell-cell adhesion, which will help to definitely remove the cell. And in a way, the description of cell extrusion is very reminiscent of the dominant view of epithelial morphogenesis, which has been dominated by two main factors. On one hand, there is the contractile forces coming from actomyosin that will tend to reduce the size of cell-cell junction. And then you have adhesive force that tend to stabilize cell-cell junction and make them longer, basically. And if you know these two parameters, in a way, you're supposed to be able to predict the cell shape and the evolution of cell shape over time. Of course, I'm a bit simplistic here, but that's, let's say, the main way to interpret most of the morphogenetic process. Now, the reason I introduced this is because by studying cell extrusion in the notum, we bump into things that didn't quite match this dominant view, which were a bit surprising. So let me explain you why. So here we have movies from the pupal notum of cells which are extruding with cadherin, and here with myosin 2, the molecular motor. And so what Alexis did is systematically tracking those cells and measuring the evolution of those key parameters. So here, this is average of many cells aligned temporally here. You have the diminution of cell perimeter. And when it comes to cadherin here, there is a progressive increase of cadherin, which means that the disassembly of cadherin have to occur pretty late in the process in the pupal notum. But what is more striking to us is the myosin 2 behavior, because we see a late accumulation of myosin 2 here, which corresponds to the formation of this cable, which was described in our system. But there is a 20-minute phase, but there is a significant reduction of perimeter area where we don't see any change of myosin 2 quantity or dynamics. And actually, if you even look at the single cell level, which I will show you now, so this is a single cell here, we have this kind of pulsatile variation of the perimeter, which match very well with this pulsatile activity of myosin 2, which has been described in many systems. But more strikingly, there is this phase where all of a sudden the cells start to constrict, and there is very little change in the dynamics of myosin 2. So Alexis did many quantification, quantifying the frequency of the pulse, the duration of the pulse, the amplitude of the pulse, no change at all. And actually, this is reflected by this parameter, which we call the contraction yield, which tells you how much a given pulse of myosin 2 deformed the cell. And actually, you see that there is a change of the regime at the onset of extrusion, where with the same quantity of myosin 2, you start to deform much more the cell and then start to constrict the cell. And so this is very puzzling because that suggests that something else than myosin 2 activation or myosin 2 recruitment have to be responsible for this change of cell shape. And actually, we also don't see any increase of Rho activity until the late phase of the process here. So what are these other factors? And to get a little bit of intuition, we actually went to a vertex model, which is the work of Alexis, another postdoc in the lab, not the same Alexis. And if I have to describe the vertex model in a very simplistic way, so you have an energy function, and the shape of cells is dominated by two main factors, a factor related to the area of cells, the area elasticity. So you have a kind of resting area. If you go below this area, the cell will tend to expand its area. If you go above this area, you will tend to go back to the steady state area. And there is line tension, which apply constraint at the level of junction, which tend to minimize the perimeter and the size of the junction, and which is a product of contractility and cell adhesion. Now, what we did is not really simulating the process, but try to get an intuition of what could recapitulate the kind of deformation that we see in the pupal notum. So this is very crude simulation here. What we do here, we increase line tension in the blue cell, and here we reduce the resting area of the blue cells, which are two different ways to trigger a constriction of the cell. And as you can see qualitatively, this is a bit different because in that case, you see that the cells become very regular. They tend to round up, which is quite expected because line tension imposes a huge constraint on the perimeter. So the cell will tend to become more circular. While here, interestingly, the cell constrict, but you can have cells which get very much elongated and very anisotropic before being expelled from the tissue layer. And this is quantified here for different set of parameters with the circularity parameter. When we increase line tension, the circularity increase. When we reduce resting area, on the contrary, the circularity tend to decrease in those cells. And what is interesting is if you look at cell extrusion process that rely on the formation of an actomyosin cable, this perfectly match with this because we see an increase of circularity. However, it's different in the pupal notum, because in the pupal notum, when the cells start to constrict, there is rather a slight decrease of circularity, which is followed at the very late step with an increase of circularity, pretty much to the moment where we see the formation of this actomyosin cable, which suggests that here we might be in a slightly different regime, where somehow the compressibility of cell is modulated at the onset of cell extrusion. So what is then driving this change of cell shape? That was the big question. So we tested many parameters, which were not all satisfactory. For instance, we tried the cell volume, because that would be one essential way to change the compressibility of cell, and caspase can change the cell volume, but there is no significant change of cell volume during the first half an hour of cell extrusion, actually. We looked at extracellular matrix, no change of extracellular matrix. We looked at actin organization, no obvious change, no change of the turnover of actin as well, until we bump into microtubules, another component of the cytoskeleton. So microtubules are interesting here because they mostly accumulate in the medioapical region of the cell here that you can see. And what Alexei noticed actually is that at the onset of extrusion, there seems to be a depletion of the microtubule from the extruding cell, while at the late phase, there is also an accumulation in the neighbor. So this kind of marker is a bit noisy. I can show you some snapshot. At the onset of cell constriction, we see a depletion of microtubule, which perfectly correlate with the onset of cell constriction, which is based on several cells here. And there is no lag time between the two. This is a cross-correlation analysis. When the cells start to constrict, this is perfectly concomitant with a decrease of microtubule, which seems to occur throughout the cells. It's not just restricted to the apical area. We see a global diminution of microtubule in the cell. So that's very interesting because that's the only marker that correlates perfectly with the onset of cell deformation here, which could suggest that maybe microtubule could somehow stabilize cell apical shape. And once you deplete microtubule, boom, you trigger an initiation of cell extrusion. So to test this, we first test whether indeed microtubule could change cell apical area. And we did several experiments. I will just show you one. So it's a rather crude one, actually, but that's quite informative. So we inject cosimid in the pupae. That's not so easy to do. This will depolymerize microtubule. Then we wait a bit. And then locally, we shine blue light on a local region here, which will tend to destroy the drug and repolymerize microtubule. So it's a local repolymerization of microtubule. And this is what you see here. There is a progressive increase of this EB1 marker because microtubule are coming back here. And I will just remove my pointer 2 seconds just to show you, if I manage. You see very clearly the change of cell shape between the beginning and the end. Boom, you see this kind of increase of cell apical area, which you can also see here, comparing the beginning and the end, where there is a quite significant increase of cell apical area on less than 2 minutes here, which you never see in all the other conditions. Could it be a controlled region here or a region where we use the same UV exposition without drug? So that means that when we repolymerize microtubule, in less than 2 minutes, we can increase significantly the cell apical area. And we did the reverse experiment. If we locally partially deplete microtubule, we don't manage to have a full depletion here. We have a transient reduction of the cell apical area. So the next question is, what's the link between microtubule and caspases? And so the first thing we did is a quite drastic experiment. We used this optodrug, which activates caspase ectopically, and see whether this could affect microtubule. And as expected, when we activate optodrug in clone, there is a depletion of microtubule, which is then concomitant with the initiation of cell extrusion. Nothing surprising so far. What is more interesting now is you can actually play with the epistasis of the pathway. We can activate caspase 9 with optodrug, but inhibit at the same time the most downstream caspase here using this P35 factor. And in that case, we have a very interesting behavior because the cell here in green will start to constrict very, very slowly, but they will not manage to extrude before hours, actually. And we don't see anymore the depletion of microtubule. This is what you see here. We even see a slight accumulation of microtubule in those cells. So basically, when we block the most downstream caspase, there is some constriction, although it's very, very slow. And there is indeed an accumulation of actomyosin, but there is no depletion of microtubule. And you quite drastically affect the speed of cell extrusion because normally cell extrusion takes place in less than 20 minutes here. So that suggests two things. It suggests that the depletion of microtubule is downstream of execution of caspase. And there is a good correlation between the impairment of cell constriction and the stabilization of microtubule. So the last piece of data, I promise, and then I will finish. So then what is the link between cell extrusion and microtubule? Can we actually somehow modulate the rate of extrusion despite the activity or not of caspase activity? And so I will just show you one result here. So what we do is we block caspase activation in clones, which you see here in magenta. So those guys will not activate caspase. And then we track systematically cell extrusion. So in the control situation, all extrusion which are marked in white here occurs outside the clone because the clone is resistant for caspase activation. If you do the same experiment, but now inject cosimi to deplete microtubule, then you start to see a lot of cell extrusion occurring also within the clone. And I will not show it to you, but we use a live marker of caspase, and we have close to 60% of the cell that extrude without any sign of caspase activation, which is something that we never see in the wild type. In the wild type, there is systematically caspase activation and then cell extrusion. And this is quantified here. In a normal situation, there is almost no extrusion in the clone. If we deplete microtubule, you restore cell extrusion in the clone, although you don't go back to the wild type level, which is quite obvious because there are other targets of caspase which must be required for cell extrusion here. But still, this suggests that the depletion of microtubule by caspase must be an important rate limiting step of cell extrusion because we can rescue cell extrusion by depleting microtubule. So to conclude, and I was a bit long, I'm sorry about that, we end up with the following model, which you can check because we put that recently on bioRxiv, where we have somehow actomyosin, which is there to constrict the cells, which is directly or indirectly, we don't know, stabilized by microtubule. Then upon effector caspase activation, this will deplete microtubule throughout the cells, which will initiate the constriction of cells without any change of actomyosin concentration of dynamics. And it's only later on, after 20 minutes, that we see a strong increase of Rho activity and actomyosin that will form a ring and terminate the extrusion process. And it's very interesting for several reasons here, because first, it's one of the few results where we see a clear link between microtubule and the stabilization of epithelial cell shape, which doesn't seem to go through a regulation of myosin 2 or Rho, because most of the time in morphogenesis, microtubule can directly modulate Rho activity, and that's how we interpret the function in morphogenesis. But here, there seems to have a more direct effect on the stabilization of shape. And there are a few papers that came out recently that goes exactly along the same line. Then the other thing is that it suggests that the depletion of microtubule by caspase is an important rate limiting step of extrusion. And if I have to make one step further and be a bit speculative here, so in the tissue, we have many cells which activate caspase and do not die. And actually, we know that once the cells start to enter in extrusion, there is no way back. All the cells that start extrusion in our tissue, they will die for sure. And because microtubule depletion is the most early step of cell extrusion that we could characterize so far downstream of caspase, it could very well be that's one of the key points that define the entry point in apoptosis and the definite engagement in apoptosis. So this is something that we are trying to test right now, and also trying to see if by changing the relative stability of microtubule, we could somehow prime the cell more or less for death engagement downstream of caspase activity. So I will finish here, and I will just again thank the people involved in the work. So this was mostly work from Leo, who now is a CNRS position, so permanent position in the lab. Alexis, a PhD student in his 4th year of studies, also with the help of Florence, a technician that did a lot of construct, including the optogenetic construct. And there was also the contribution of Alexis, who did the vertex model of we have another system and another project, which I didn't have time to speak about. And also we got some help from Angela, a mathematician in Pasteur, for the statistical analysis of cell death. And I will stop sharing my screen here. I've been talking fast. I hope we still have time for some questions.
Thanks, Romain. Nice talk. Any question? Can you hand up first? Roberto?
Yeah, hello. Very nice talk. So I am not sure if I understood correctly the last model. I mean, you are suggesting that this ensemble of microtubule is a driving force for the extrusion. But you are not saying that you don't require this actin contractility.
Exactly.
I mean, probably you require both.
Yes.
I guess if you don't have actomyosin contractility, you don't have extrusion, even if you don't have microtubules. Have you tried to do I mean, to prove that really, I guess the way in which I imagine is that you have a balance between the force generated by the actomyosin contractility and the opposite force by the presence of the microtubules there. So have you tried to if that is the model, have you tried to play with this, play with the levels of actomyosin contractility and with the levels of microtubules to see really if there is a balance which is required?
So yeah, so it's a very good point. So first, you're totally right. So first, to be clear, microtubule, in a way, if they do anything, they stabilize. So what you need is to deplete them to be permissive for deformation in a way. Now, to be really clear, we cannot argue it's a direct mechanical effect of microtubule. It's very hard to prove in our system. So it could very well be an indirect effect, which could be through, for instance, the organization of actomycin, which become more efficient at contracting, for instance. So this we could not detect. We have limitation regarding what we can detect and assess. So what you suggest is very nice, but the fact is we lack a bit of tools to do some kind of fine tuning of the level of stability of microtubule. And same for actomycin. Actually, surprisingly, if you deplete rho kinase, for instance, it's not a full depletion. Cells still manage to extrude somehow. The regime is a bit different, but they still manage, although it's not a full depletion, so they may still be able to make a bit of contractility. The thing is, what happens at some point, which is a bit what we see when we trigger extrusion of cells in clusters with optodronc, at some point you start to disassemble cell-cell adhesion as well. Normally this comes very late, but what I suspect is that if you slow down too much the process, then at some point caspase will start to disassemble everything, including cell-cell adhesion, and somehow you will have some sort of aberrant cell extrusion that still manage to keep the cell out, but maybe there will be some gap formation. So it's, but it's something we have to test. Yeah.
Sorry, do you know, I mean, just a short additional question. Do you know if when the cell depolymerize the microtubules, do they become softer?
So that we would love to be able to measure, but we can't here because, you know, the contrary to other system, we have a cuticle, so we can't really directly probe the mechanical property. That's why we don't want to make big claim about the mechanical property of cells, but that's what we would expect maybe. Yeah, this we can't really measure directly. We are limited on this.
Yeah. Okay, thank you.
Next, Guillaume.
Hi, Romain. A very nice talk. So actually, I was curious about your statement that that cells that were at the boundary between, let's say, winners and losers, those cells would be most likely to die because they would rarely receive any pro-survival ERK signals. So I mean, surely this is something that like, for example, in mechanical competition, I thought that was something that people had looked at, and they felt that there wasn't any clear relationship between the position of a cell at the interface between the two, you know, the winners and the losers. And so I was wondering if you could maybe comment on that.
Yeah. So obviously, it comes back to the slide I showed at the beginning. There are different modes of cell competition, and what we see, for instance, in the context of like what you see in the mechanical competition, there is no strict need to be in contact with the other cell type. This for sure we see as well. What I was alluding to here, and it's speculative, it's that just having this kind of caspase resistant clones may be sufficient to see a local increase of cell death. I don't want to say that it's going to explain every single cell competition scenario we've seen so far, but I was just mentioning that this simple difference could already reproduce part of the signature that we found in certain cell competition scenario, like the MIC or the minute competition. It's exactly what we see. There is an increase of apoptosis at the boundary. Now, of course, most likely there are plenty of other things at play. I'm kind of adding another layer of complexity here, but yeah, but it doesn't. Yeah, for sure, the mechanical competition, including what we see in our system, you don't need a contact. You don't need a contact, so it cannot be explained by this.
But in terms of the ERK signaling, if you look at the boundary between you know, a clone that would be a loser in a mechanical competition and one that would be a winner, do you also see that, you know, do you have that difference in ERK signaling that occurs?
So that go back to the, so when we do this kind of, when we look at ERK in those more massive competition scenario, the thing is there is a global change of ERK in the clone. So so I guess the global output at the end is a convolution of a kind of global modulation of ERK plus the change of these dynamics, which makes things a bit complicated, I guess. And we haven't tried to analyze. I guess the difficulty and that's one of the experimental difficulty, we use mostly clone tool to analyze this thing, and the clone is completely viable from one experiment to the other. What I'm talking about is a very subtle bias. So I hope, but I'm not sure we'll manage to detect it experimentally, because of course the clone is never exactly the same place, exactly the same shape. So we need to find a more reproducible thing, maybe using some kind of optogenetic clone induction now that it's possible. Yeah. So yeah, no, you're totally right. I think the thing is it's the same pathway in the end. So then the reason is, how do you kind of, you know, integrate all this thing is an open question.
Okay. Thank you.
Thanks.
Okay, next, Mark.
Hi, Romain.
Hi, Mark.
I really enjoyed your talk. I had a couple of questions about the first part. So when you described this local inhibition of cell death, So that reminded me, and I wondered if you had any comment on that, of what's seen around wounds. You also see something similar around wounds where there's a local inhibition of cell death, and if you thought it was the same mechanism operating. And then the other thing that's also related and also reminds me is in essentially compensatory proliferation mechanisms. So dying cells are known to secrete signals that promote proliferation of neighboring cells to make up for their loss. And I was wondering if you thought maybe, which as far as I know, EGF has not been characterized as one of the compensatory proliferation signals, but if you thought maybe that could be the case.
Yeah, so that's a very relevant question actually, and so yeah, the link between wound healing, I guess there could be, although I don't know if it's exactly the same process, because there you generate a big gap, there is some kind of leakage and big calcium wave, for instance. So in our system, for instance, we don't see any calcium activity, and I don't think we don't have this super nice time resolve GNK reporter, but we don't see obvious GNK activity, for instance. So I'm not sure it's exactly like a wound response, although there might be some commonalities in the signal. Now regarding compensatory proliferation, it's a very good point. The thing is, we studied so far this in the notum where the cell divisions are very stereotypical. So it's a bit different from the wing disc, for instance. And if anything, it's not obvious. We haven't seen a clear effect on proliferation. But now it opens an interesting question, because that's something that we are starting to study in the lab. The thing is, compensatory proliferation have been very much studied in case of drastic cell death induction, or I mean, I guess, like a massive induction in large clones, or blocking the apoptotic pathway very downstream. I think whether when you have only one cell dying here and there is a significant impact on the proliferation of the neighbors is still an open question, I think. But and that's something that I would love to be able to address actually, because that would be interesting. But yeah, maybe we'll find that it's the same, or maybe you need a certain quantity to see this kind of classic compensatory proliferation thing. Yeah. And
in terms of what's activating EGF receptor, I guess you've probably thought of this, but the obvious ligand is vein, because it's membrane tethered.
Yeah,
I was wondering if you looked at vein expression.
Yeah, so that's you're touching over a question which is tricky. So we try to defeat one by one those ligands. So vein didn't give an obvious defect, but the thing is, none of the ligand depletion give a super strong phenotype. It's only when we deplete EGFR, which suggests that there might be some kind of redundancy. I should say the most interesting one we see is Spitz. Where when we globally deplete Spitz, we see an effect on ER quite clearly in the notum. But then for sure, if you deplete Spitz in the dying cells, you can still see the process. So it doesn't seem to be, you don't need the secretion from the dying cells. What is a bit less easy to understand is if we deplete in the neighboring cell Spitz, there seems to be a slight reduction of the responsive of the cells in the ERK signal, which could be a kind of a autocrine activation indeed. But it's not like a complete abrogation. It's a significant decrease, but we don't completely abolish the pulse. So yeah, maybe we should look more into vein to see if this could participate. Yeah, it's a big question. Actually, what we've been trying many things one by one, excluding the system, the model. What we are checking now is whether there is some kind of dynamic relocalization of EGFR depending on the deformation of cells. There seems to be something on this line that could be important, but we have to, we had to make some tag version of EGFR, and you always have to generate new tools because there is not so much, as you know, on the ERK signaling pathway in flies, surprisingly. So yeah.
Cool. Thanks.
Thanks.
Okay. Thanks, Romain. We're a bit shortage of time, so a bit busy day. Expected to meet other people as well. So thank you, Romain. It was really fantastic talk, and then I'm looking forward to seeing you virtually later on.
All right. Thank you for joining virtually, and don't hesitate to shoot an email if you have a question.